Hi, my name is Konrad Krawczyk, and today I'm going to talk about antibody sequence clustering and how this can be used for therapeutic antibody discovery. So altogether, we have two major experimental ways of discovering antibodies these days. One is phage display, the other one is immunization. In both of those techniques, we can use NGS to get a bunch of sequences from which we have to select a set that are binders and that are devoid of any obvious computational liabilities. One more thing that is important about this, like in a smaller set that you're identifying at the very end, you want, so to speak, hedge your bets with it. Yeah. So you do not want, if you select 50 sequences, you do not want those 50 sequences to be the same thing. Yeah. But the same thing can be, it actually is so, the sequences are so similar to one another that it is kind of like picking the same sequence. Yeah. So that's why not only the binder selection, which comes from grouping, the liability removal, but also the diversity step at the very end is actually quite crucial. So let's go superficially through both methods right now. Firstly, phage display. Here you start with an antigen immobilized on a plate. You have your antibody library expressed in a phage, and you have the antibodies on their surface. You are panning the library onto the antigen. You're washing off whatever doesn't bind, and whoever bound, you are using them to amplify them for another round, and you're redoing it over and over again. The over and over again, it's usually several rounds. And as you can imagine, whoever bound and is then amplified is going to increase their numbers, with each amplification. Yeah. So like when you start off from a very, very diverse library, then you are actually reducing this diversity and you want to capture the ones that actually took over most of the diversity. This is where the NGS comes in because you want to sequence those antibodies. So you want to identify 100% or at least fuzzy CDRH3 100% clusters. And you want to be able to say which of the sequences actually increased in their overall frequency from one round to the other. Yeah, because these should be the stronger binders. So when I had one sequence that increased its frequency quite a lot from round 1 to 2, then this is something that the clustering obviously would allow me to find out. Yeah, because then I know the proportion in round 2 of this sequence and I can compare it to the proportion from round 1. Yeah, it should be bigger. Then to antibody immunization. So here you start with an antigen, you inject the animal with a said antigen, and then all the vertebrates are going to do what we usually do, which is develop a lot of antibodies against the said antigen. So then you collect cells that are producing antibodies that should be specific for the said antigen, such as plasma cells, and you then try to capture the biology of it. Yeah, so you are identifying the so-called clonal families. Each clonal family should be a set of sequences that diversified from the initial binder. Yeah, so on the left-hand side of each of the trees, you have let's say, the parent cell, and then as the sequence matures, it is farther and farther away from this germline cell. So here, what you want to capture from clustering is the so-called clonal family. It can be defined in various ways, but one of the most typical ones is that it needs to have the same gene and it has to have the same CDRH3 length, and then you are allowing certain leeway in your CDRH3 identity. So for instance, like 80, but 90, 95%. Yeah. Really depends on your definition. What you do want to select from each of those clonal families, so these are your clusters here, are sequences that, according to you, are either more or less diversified. The more towards the right, you're more diversified, you should be a stronger binder. Towards the left, you should be less diversified, so a weaker binder. So These are the two ways that grouping of the sequences can be used to address binder selection either from phage display or immunization. So another step that I mentioned is the early filtering. So some libraries actually already have this step um, coded in. So for instance, from Specifica, you have libraries that are devoid of liabilities. Yeah. But still, there is a quite a wide variety of tools these days that allow you to look at in your sequences, whether this is at the clustering stage or at a candidate selection stage, tell you, are there any liabilities? Are there any obvious developability issues with them, but predicted computationally? And of course, you can compare them to your own assay data. Yeah, that's if you already have that. So this allows you to, at the very least, be able to navigate in which clusters might have better or worse properties according to certain computational features. And I would like to say one thing about this filtering. So currently, at least in the public domain, there is not a silver bullet that is going to tell you whether a given sequence, just by the virtue of its preclinical data, is definitely going to be a success in the clinic or a dud. Yeah. But this is not to say that they are not useful, because at the very least they are telling you if there is something quite obviously wrong with your sequence. Yeah, that's not something that you would like to find out at the clinical trial stage. Then going towards the diversity. So here, when we start with a whole bunch of sequences, whether we are doing the enrichment from phage or we are doing the clonal selections from immunization, you end up with a smaller amount of sequences. I would like to have those sequences diverse. Yeah, this is a very let's say ambiguous term, because if I say that those sequences are diverse in terms of sequence, if they have only one amino acid difference, then yes, they are diverse, but it is quite likely that they actually have very, very similar properties to one another. Yeah. And I would actually need to cover different bases to increase my chances that the sequence actually has a good amount of properties. Yeah. Otherwise, I'm checking pretty much the same thing. So one way of looking at the diversity problem, at least this helped me because it's structural, is literally looking at the crystal structures and predictions of those. So imagine on the left that the blue circles are antibodies and the red circles are the sequence clusters of those. If we use a very simple example of clinical classes of antibody CDRs, then it is quite easy to envisage that two different sequence clusters could actually fall within one structural one. So right now, this is up to me as an engineer to decide whether I would like to collapse those two red sequence clusters into one, because otherwise, they're the same diversity according to the structure. Yeah. So this is something that actually we checked whether if you use any of those other clustering algorithms, are they actually better at separating the binders and non-binders. So we have the old clustering methods, sequence clustering and clonotype clustering. So obviously clonotype clustering is a subset of sequence clustering. And we have the paratope. So whenever you have a paratope prediction and your sequences agree on which residues are the paratopes, then they are in the same cluster. Structural clustering, like canonical classes. If you are a very similar sequence structural shape, then you should be within the same group. And the most abstract one, so embedding clustering, we used language models here to learn the representation of antibodies that should be more efficient than just the sequence representation. And when you are comparing the representations of the embeddings vectors of those sequences, then in theory, this should give you yet another dimension to look at the diversity of those. So are those actually better? Well, the short answer is no. When we checked, and you can look in the paper, but you have uh, a pure curve here that shows that essentially they just all fall within the ballpark of one another. Yeah. So are they actually not useful? So should you go for the least computationally expensive one? No, because they actually offer very diverse picks. Yeah. So even though they one is not better than the other. It's separating binders and non-binders. One is actually being able to tell you that those are more diverse than others because they, for instance, have a different structure or they are in a different embedding group. So at the very least, it gives you another dimension of looking at how to actually uh, select your final picks, not only by the virtue of sequence. So from all of that, a pipeline starts to emerge. So I'm going to address how you can stitch it up using publicly available tools. So the initial step, sequencing, you cannot skip that, yeah? But whenever you get to the NGS stage, you can start using tools such as Anarchy or IGBLAST to process your sequences for QC, germline annotation, and numbering. Then at the clustering stage, there are tools such as MMSEX or CD-HIT that can group your sequences, but mostly using sequence. Then in terms of filtering, you need to get a whole number of different tools, and I'm definitely not going to list all of them here, but you can find quite a lot of them on the subpred website, and there are tools such as Prom, BioFi, and so on that do naturalness. Then final diversity selections and the final selections, there are many pieces of software that do this, or at least visualize your sequences. But from what I see, there is still quite a lot of heavy Excel use. Yeah. So, and of course, there are open pieces of software such as Incantation that actually can cover a lot of those bases here. So now, what can you, is there a platform that actually it does all of that? Yes, there are several of them. They are the enterprise platforms. You need to pay some money for quite a lot of them. We offer a lot of this functionality for free. And I'm just going to go through an example of what we do. So you can submit your sequences to us, and this is either install it on your own premises or a cloud platform, and we are going to number your sequences and perform germline annotations using Riot. In terms of clustering, you can look up the CLAP paper that I talked about earlier, but we do all kinds of different clustering methods. Filtering, we do have our own methods and re-implementations of the publicly available ones for developability, naturalness, and liabilities. And the final selections of diversity are within our GUI. So you can see the multiple sequence alignments together with the different annotations. So one thing I would say is that having this all in one place at the very least gives you a convenience, yeah, of it rather than having in different teams and different people are responsible for a lot of very disparate scripts. So this is it. I hope that it was informative, and thank you very much for listening.